Chapter 5 Antioxidant property of hypophyraminoids and its role in the management of neurodegenerative disorders. Oxidative stress plays an important role in neurodegenerative disorders. The term oxidative stress refers to a state in which free radicals and their products are in excess and causes metabolic imbalances. This imbalance can occur as a result of increased free radical production or a decrease in antioxidant defenses. The free radicals, particularly reactive oxygen species, ROS, are capable of reacting with lipids, proteins, nucleic acids, and other biomolecules and altering their structure and function. Oxidative stress can lead to alterations in cells with an accumulation of oxidized products such as aldehydes and isoprostanes from lipid peroxidation. Protein carbonyls oxidize protein, which in turn form base adducts with DNA, thus serves as markers of oxidative damage. Marcus Berry 1999, Beckman et al. 1998. The membrane lipids in brain contain high level of polyunsaturated fatty acid side chains, which are prone to free radical attack. Brain has been shown to contain mild to moderate level of enzymes such as catalase, superoxide dismutase, and glutathione peroxidase, which plays an important role in the metabolism of ROS. Cohen 1988, Markland et al. 1982. The presence of iron in globus pallidus and substantia nigra also contributes to the production of ROS. Jaleska et al. 1989. Halliwell Gutteridge 1985, 1989, Joseph 1992, Jenna 1994, Frolic Readerer 1995, Marcus Berry 1997. Transition metals, such as iron, copper and manganese has been associated with increased free radical production through the Fenton reaction, Packer 1995. Extensive research indicates the role of aluminium in a variety of neurodegenerative disorders including Alzheimer's disease, which has been found to be associated with the formation of ROS. Aluminosilicates have also been identified at the cores of senile plaques, and aluminium has been found within neurons bearing neurofibrillary tangles. Aluminium has been shown to be involved in inflammatory response mediated by interleukins and other inflammatory cytokines and to exacerbate events associated with AD, Campbell 2002. Increased level of aluminium is an important catalyst for free radical generation in the brain of AD patients. Marcus Berry Ehman 1993.
Marcus Berry 1997. Moreover, genetic abnormalities of transferrin C associated with defective binding of iron and aluminium are associated with higher rate of AD, Van Rensburg et al. 1995. Studies carried out in AD patients have shown an increase in the levels of antioxidant enzymes such as superoxide activities, Subarov et al. 1990. However, there is a marked heterogeneity in the degree of response in the different brain areas, Bolish and Leon 1994. For example, high levels of immunoreactivity to catalase have been found in the neuron associated with neurofibrillary tangles of Alzheimer's patients, Papala et al. 1994. Chen et al. 1994, have found that catalase activity is significantly higher in the cerebellum, frontal cortex and temporal cortex of Alzheimer's patients. There are known antioxidants which have shown significant potential role in the protection of cerebral cells injuries induced by oxidative stress. It is necessary to find out some plant-based safe and effective antioxidants to prevent or to slow down the process of diseases related to neuronal degeneration. Lipid peroxidation is a predominant means of neuronal degeneration in Alzheimer's disease, Subarov et al. 1990. In this mechanism, hydroxyl radicals strip of hydrogen atoms form polyunsaturated fatty acid side chain of membrane phospholipids. The membrane phospholipids are converted to peroxy radical and are able to propagate the steps repeatedly, thereby destroying the composition of the phospholipids membrane and altering its properties. This process results in the disruption of membrane function, decreasing its fluidity, and permits the unregulated passage of ions such as calcium to form the cell, thereby creating the potential for lysis. All these events ultimately results in reducing the efficiency and function of the neurotransmitter system and initiate the disease. Halliwell 1989, Rafalowska et al. 1989, Franceschi et al. 1996. Studies have shown a relationship between elevated lipid peroxidation in the frontal cortex of brain in Alzheimer's disease. Subarov et al. 1990, Marksbury and Carney 1999. Enhanced lipid peroxidation is recorded in specific areas of the brain in post-mortem studies. Lovell et al. 1995. Trace elements such as iron, ferritin, aluminium, mercury and copper stimulate the lipid peroxidation in neurodegeneration of Alzheimer's disease. Quinlan et al. 1988, Kennard et al. 1996. Catabolism of dopamine also serves as a significant source of free radical generation in Alzheimer's disease. Cohen and Spina 1989. Other possible mechanisms for the causation of Alzheimer's disease include the production and accumulation of glutamate hydroxyl radical and overproduction of superoxide dismutase, leading to the accumulation of hydroxyl radical produced from the conversion of hydrogen peroxide. Bell et al. 1992, Vinken and Broin 1977. The hydrogen peroxide is produced in response to aggregation of amyloid A protein in the neurotic plaque in the progression of Alzheimer's disorder. Bell et al. 1992. Alzheimer's disease is characterized by the deposition of amyloid A peptide, AA, within the neocortex that is associated with neuronal demise and oxidative stress. The cause of the neuronal loss in Alzheimer's disease is unclear, but increased oxidative stress from excessive free radical generation plays an important role in etiology of the disease. Smith et al. 1997, Bush 2000, Sayer et al. 2000. One of the major potential sources of free radical production in the brain is from the transition of metals such as copper and iron. Bush 2000, Sayer et al. 2000. In recent years, antioxidants have gained importance because of their potential as prophylactic and therapeutic agents in many diseases. These are the agents which scavenge free radicals and prevent the damage of lipids, proteins and DNA in neurons. In general, antioxidants include enzymes, i.e. catalase, super-oxide dismutase and glutathione peroxidase, low molecular weight compounds such as glutathione, vitamin E, retinoic acid, ascorbic acid, albumin, bilirubin, ferritin, ceruloplasmin, melatonin, uric acid, lipoic acid, minerals and phytonutrients, flavonoids and carotenoid. Antioxidants are part of diet, and fruits and vegetables are a rich source of vitamin C, carotenoids, while whole grains and high-quality vegetable oils are major sources of vitamin E. Halliwell 1994, Jacob 1995, Bloch et al. 1992, Azzi et al. 2004. Hepatic uptake and renal excretion are the major pathways for the elimination of antioxidants enzymes. Antioxidant enzymes are more potential agents in treating severe acute insults due to oxidative stress. Christophidou et al. 2006. Elevated levels of erythrocyte glutathione peroxidase, superoxide dismutase, and catalase activity are observed in patients with AD compared with control subjects. Anran et al. 1986, Perrin et al. 1990. An elevated level of superoxide dismutase activity was also recorded in fibroblasts form of patients of AD. Thienhaus et al. 1986, Wiener et al. 1988, Zemlin et al. 1989, Rossetti et al. 1988, have determined inhibition of free radicals by super-oxide dismutase and by monoamine oxidase inhibitors generated in Parkinson's disease model induced by MPTP. Several epidemiological studies have shown that ingestion of vitamin A, E, or C may result in higher cognitive performance and reduced risk for developing clinical symptoms of AD. Masaki et al. 1994. The ability of vitamin E in suppressing lipid peroxidation depends on the solubility and localization in the lipid membranes of CNS. Studies by Foy et al., 1999, have demonstrated a significant depletion of plasma levels of vitamin A, C and E in Alzheimer's disease compared to controls, which supports excessive free radical activity in AD. The CoQ10 is a very potent anti-oxidant. It is observed that patients suffering from Parkinson's disease have a very low CoQ10 level, for which various lifestyle and environmental factors are responsible. Low level of CoQ10 leads to low complex 1 activity and causes deficiency of mitochondrial function, increases reactive oxygen species, ROS, formation, which further damages mitochondrial function. A supplement of CoQ10 is helpful in the prevention and management of neurodegenerative disorders, particularly Parkinson's disease, Schultz et al. 1997, 1998. Vitamin C and iron supplementation protect mitochondria from free radical damage and reduces the cognitive decline. Alzheimer's disease is characterized as a disease with reduced glutathione. Therefore, increase in glutathione level is the fundamental for the management of Alzheimer's disease. It has been reported that use of Namenda significantly reduced the functional deterioration in Alzheimer's disease cases, McCaughey et al. 1998. A correlation between memory function and vitamin C in the blood of healthy volunteers aged 60 or above is found by Goodwin et al. 1983. Accordingly, Perry et al. 1997, have found a positive association between memory performance with A-carotene and vitamin C levels. Vitamin E is a lipid-soluble antioxidant that is very effective in suppressing membrane lipid peroxidation. Another study showed that vitamin E could preserve learning and memory function, which gradually declines during aging, Sochi et al. 1995, Xia et al. 1993. Clinical trials of vitamin E in Alzheimer's disease patients have yielded positive results, with patients receiving this antioxidant exhibiting slow progression of disease compared with those receiving placebo, Grundmann 2000. Vitamin E may counteract the effects of aging and neurodegenerative disorders by suppressing membrane lipid peroxidation and thereby preserving membrane transporter function and stabilizing cellular ion homeostasis. Lipoic acid is an endogenous disulfide compound that is present in small amount in most animal cells, where it functions as a coenzyme in the ketoglutarate dehydrogenase and pyruvate dehydrogenase enzyme complexes, Marquet et al. 2001. The antioxidant and cytoprotective actions of lipoic acid have been documented in the variety of cell culture and animal models of age-related disease, McCarty 2001. Lipoic acid and the related compound dihydrolipoate are